So my name is Henrik. I'm the Biosafety Coordinator here at the university, and I'm going to talk about the biosafety legislation and the permits you might need. But first off, I'm getting the feeling here that you're more into perhaps larger organisms than we normally think about when we're thinking about biosafety, right? Not so much microorganisms, perhaps a little bit of parasites somewhere. Yeah, okay, good. Then I'll skip through that bit quite quickly and focus on the things which might be more relevant for you. Good. So this is sort of what we think about when we talk about biosafety. Mainly, it's of course the things in the middle, virus, bacteria, fungi, microorganisms like that. But it also includes blood and tissue, animals, plants that are genetically engineered in some way, or perhaps invasive also, then that could also be an issue. And of course lab waste from all of this. So it ranges from like the nanometer up to the meter size, depending on what you're working with. And in order to work with these, and then in particular microorganisms, they've been grouped into 4 risk classes so that we can build different laboratories for different needs. And I think you will mainly be at level 1, although I imagine level 2 will sometimes happen actually, especially when you're working with blood and tissue and so on. So I'm going to go through these classes so that you have some background to where you need to work. So this is then called risk class and containment level in British English. Some of you might be more familiar with the American English, where they say BSL, biosafety level. The French say P, we in Sweden say risk class och skyddsnivå. But the gradient is the same, 1 through 4. 1 is the least dangerous, does not cause disease. 4 is the most dangerous. So the shortest possible definition of risk class 1, or risk class 1 microorganism would then be that it does not cause disease in a healthy human being. Baker's yeast, which you see here, is a good example. We have it in many labs, and we don't need too much in terms of safety, right? Although, I should stress, if they are genetically modified, which they often are, that's why we use them in the labs, then they are not allowed to escape the lab. And in the EU, anything which is genetically modified must stay in the lab or be destroyed. Yep. So this is not a pathogen, because it cannot make you sick. This is a pathogen, risk class 2. That's the first time we call it a pathogen. It can cause curable or transient disease. Chickenpox, you see here, most of us have had it. Hopefully we have lifelong immunity. Containment level, well, now you do need a little bit more. You should have a door to the lab at least, and you're probably wearing lab coat and gloves. It might be a good idea to have an autoclave in the lab. And also these safety cabinets. If you're working with something which is airborne by nature, or if you're creating aerosols, then you need one of those. Here I should also mention that these risk classes are based on the disease they cause in humans. But you should also consider the environment in animals. I make the example with Newcastle disease virus, which could give us a mild cold, if even that, but for the birds it can be very deadly. So if you're going to work with that one, well, you want to add some more protective measures in case it escapes the lab, right? Yeah. We don't always think about this, and the legislation is quite strict and clear when it comes to humans, and sometimes we forget the animals. Yeah, so that's risk class 2, and it doesn't look too bad, right? But this is also risk class 2. These terrible infections where you have the multi-drug resistant bacteria, they're also risk class 2. And there are many labs where they do research on these particular strains because they want to find new antibiotics and also prolong the life of the antibiotics that we do have. Yeah. So bear that in mind. Risk class 2, it is not completely benign, put it that way. Then risk class 3, this is serious illness. You see the HIV virus, and the containment level, well, of course you need more now. You probably also, before your lab, you probably have a separate room where you change, and in the lab you're not just wearing gloves and lab coat, you probably have a disposable gown on top of your clothing, and you're probably wearing double gloves so that there's distance which tends to open up between your glove and your lab coat. You make sure that that is covered at all times. And also, this shouldn't be accessible to everyone. With a key card like this, at least at Uppsala University, you can access many level 2 labs. And that's well and good. But level 3, that is only designated access. Those people who actually need to be there have access, other people no. And then the last class is level 4. Ebola is a good example. You only have viruses in this risk class so far. Containment level is essentially a separate building, and you have these high-pressure suits and you shower in and out and such. Very expensive to build and operate these labs. And of course, the reason why we're not always working at this level is because it's very difficult and it's horrendously expensive. I would argue that if you, with the increase of a risk class, you increase the cost tenfold almost. So I gave you the one-sentence definition. There is, of course, a full legal definition. We have this in Sweden. It's Arbetsmiljöverket who handle these legislations, and it's the tillsynsmyndighet, whatever that is in English. And I made a quick co-pilot translation for this, because unfortunately the legislation is only available in Swedish. But apart from what I said, the one-sentence summary, they also include things like how easily does a microorganism spread? What happens to society if it gets out? Is the vaccine available? All these things go into determining what risk class it is. It might be relevant for some of you if you're going to sequence some new, the next exciting pandemic. You want to get that and sequence that one and work with that in your lab. Well, then you need to go to this to figure out which risk class it is. SARS-CoV-2, of course, wasn't on this list, but they figured it out and put it as number 3. They also had in this legislation a guide to a whole bunch of pathogens, and then you can go in and see what level you should work at. So let's have a practical exercise. If you take up your cell phones and put it on this QR code, and then tell me E. coli, which risk class is it? I think most of us have worked with E. coli, right? Yeah. At some core strategies. Depends. Yeah.
In the spring.
Yeah. So you do protocol? You do it. So has anyone found it? What risk class does it take? Anyone found it? 2. 2. Anything else? 3. What's the difference between 2 and 3 then? What do they say about the number 3 stuff?
CX0157H7, but gives some specification of which strains properly. It is upper level 1.
They also say something about toxin producing there. Yeah. So it's the really nasty strains are 3, but otherwise it's 2. Has anyone worked with E. coli at the level 1 lab? Perhaps during course lab or even with students? I know I have. Yeah. This is because they also say something in Swedish, with exception for non-pathogenic strains, med undantaget icke-patogena stammar. So E. coli is funny that way because it ranges from 1 to 3, depending on which strain you're working with. Normally, when we hear E. coli, we think 12, right? And that's because most of the lab strains that we use are indeed level 1. That's why we like them as lab strains, right? And if you want to find out, you can go online and see which strain you're working with. Here I punched in E. coli DH5 alpha, and sure enough, they say it's biosafety level 1. And there are many of these online resources available. ATCC is kind of nice, although it is an American website, and sometimes the rules are a little bit different. The gradient 1 to 4 should be the same, but sometimes different government agencies in different countries can make their own determinations. For instance, dengue virus is risk class 3 in Sweden, whereas it's 2 in the US. And it can also go the other way. Humula virus is level 2 in Sweden, but it's 3 in the US. Yes, and in these legislations and guides how to work with microorganisms, they are of course always talking about the normal infection balance. But what you're doing in the lab when you're working with your microorganisms like this is usually you need to cultivate it. That's usually the way for it. You cultivate it and then you concentrate it and you extract your DNA or RNA, whatever you have. And then something which may have been dependent on contact for transmission, if you're having it in a test tube like this researcher here and you start sniffing your sample, well, maybe that's enough for it to spread through the air when you have it at that extreme concentration, which you would never find in nature. That's why we sometimes have to use a little bit more precautions in the lab than you would perhaps find in the clinical setting in hospitals. Fair enough? Good. If your work takes you in a direction where you need to work with something which might be really dangerous, Then most of the larger labs have high containment facilities. This is from our Zoonosis Science Center. So there they can handle samples of this type and maybe cultivate it and extract the DNA or RNA or send it to you, and then you can work with clean DNA or RNA. That might be good to know. So there's no real need to work in a normal lab with something that doesn't belong in a normal lab, because the bigger universities have facilities which can handle it. And you can contact your biosafety coordinator to find out how to access that. This is an overview of the legislation, which is geared mainly towards biosafety or interacts with it in some ways. You already had a look at the first one there when you found out which risk class an E. coli was. There is a separate legislation if it's genetically modified and contained in the lab. And this is done for microorganisms contained in the lab. And there is also other legislations if you're working with higher organisms which are genetically modified. And at last count, there are 8 different in Sweden. There's 8 different government agencies poking around in genetic modifications, depending on what you want to do. But if it's in the lab, it's mainly Arbetsmiljöverket. And you have some other legislation for transport and CITES and or byproducts, which I will talk a little bit about. This is, if your research takes in this direction, the government wants you to report it. So this is a guide if you need to report it or not. So if it's risk class 1 and it's wild type, you don't need to report it, right? Because let's face it, if you're eating yogurt in the morning, you're eating risk class microprocess. And you don't need to tell Arbetsmiljöverket when you're eating yogurt, so that's good. However, if you made the yogurt fluorescent, if you said a GFP class or something, then you would need to report it. Same for the risk class 2, then you need to report both wild type and if it's genetically modified. For 3 and 4, if it's wild type, you need to report it. But if it is genetically modified, then you need a special permit. It's a little bit of a more difficult process. But contact your biosafety officer before you send in these reports, and they would help you with that. And the basic idea from Brussels is that if it's genetically modified, you can't know for sure what's going on. That's why you will encounter more bureaucracy when it's genetically modified. It's just the way Brussels sees it, I'm afraid. There are other things which may influence, which you may come in contact with in your research, and those of you who are importing samples and working with different kind of samples from all over the world. One is animal byproducts. I asked co-pilots to make an illustration of the EU animal byproduct legislation, and they came up with this, which is sort of indicates where we're going. It's mainly geared towards protecting the foodstuffs that we eat. So if you have a cow which died on a farm, that shouldn't be grinded down and served to other cows, right? We want to avoid that. But unfortunately, this legislation also applies to research of sometimes, let's say you want to import serum from a mouse, and that would fall under this legislation. Some universities have made blanket permit for this, so you can contact your biosafety officer. We have it here at Uppsala, and if you need that, you can talk to me. Here I should stress, though, that this is for animal byproducts. It is not for the plants, it is not for in-betweens, as we talked about. It's not for CITES or Nagoya, and it's definitely not an ethical permit. But at least it's one permit which you don't need to apply for yourself, because we did this for you. So talk to me if you need that, and I will show you a list about my counterparts at other universities in the end, and you can contact them if you need it. Then when it comes to CITES, this legislation for the Convention on International Trade in Endangered Species of Wild Fauna and Flora. Again, I asked ChatGPT this time to make a picture illustrating it, and they came up with this. I especially like the sort of traveling bag crocodile lion thing which made on her. That's particularly nice. But it is sort of what it is. The intention was to make sure that we don't trade in endangered species and reduce their numbers. But it also applies to research. Again, unfortunately, I have to say, because it can be a bit of a wet blanket for research, just as Nagoya can be. I talked to Jordbruksverket about this because they are the government agencies which handle it, and I tried to work my way to get one of those blanket permits, but they wouldn't have it. I haven't given up yet. I will try again and see if we can do it. Olga would like it, I think. Yes. So we also asked Jordbruksverket to come here and talk about this and explain how we can apply for this in a streamlined way so that we don't run into trouble. Because it happens. Even if you do everything right with this legislation, you can still sometimes run into trouble, unfortunately. But this is, in a way, how they say on their website, Jordbruksverket, that you should do it. You go to this website called Species Plus, you type in whatever you want to work with, and they say if it's listed here as a seated species or not. But when I talk to Jordbruksverket, they say sometimes, although it's very rare, species which are on the list are not listed here. So then you have to go to the legislation yourself. And I think you will get handouts from this presentation, right? So you got to get all these QR codes sent to you. Yeah, because pretty much like in Nagoya, you don't want to get in trouble with this, because then your samples will get stuck in customs, and if they're stuck, and yeah, I can't help you with that. And I'm not sure that will happen. Does anyone here have problems with CETAs? Yeah? 4 or 5? Yeah. It happens, unfortunately. So make sure you have this squared away before you send your samples. This is then the contact person at the various universities. So you have me there, and I included a QR code for the biosafety information, which we have on our website. I went through it very quickly now because it doesn't apply to all of you, so you can go there and find out more if you need it. For Nagoya, they are contact persons for Uppsala, and also Stockholm University had special function for this. For general research ethics also, you have some contact information.